What's up, guys? Welcome to another episode of Camber. And today I'll be talking about serotonin syndrome. So, what is serotonin syndrome? Well, starting from the basics, serotonin is a neurotransmitter found in the brain which neurons use to communicate with each other, one of many different types of neurotransmitters. There also happens to be a medical condition called serotonin syndrome, where your brain, there's basically some dysfunction leading to altered amounts of serotonin in your brain, which can lead to an altered mental status, as well as the dysfunction of your autonomic nervous system, and is usually thought of as a toxidrome, or basically brought on by ingesting multiple of different types of serotonergic agents or drugs which influence the amount of serotonin that is produced in your brain. But before we talk about serotonin syndrome specifically, I think it's important that we talk about the serotonin system generally. And just to give you a roadmap of where we're going in this video, first of all, I'm just going to talk about why am I making this video right now, and is there any specific reason? And to that answer to that is yes. Secondly, as I mentioned, just talking about the serotonin system, what is it? What does it look like in the brain? All that good stuff. Then after that is where we get into the fun of discussing what does your brain look like under the influence of psilocybin, and looking at MRI data to see how its function is altered in this crazy state. And then lastly, we're going to look at serotonin syndrome, or as I mentioned, it's basically just faulty serotonin signaling in your brain and how that can happen, which I'll clue you in. It can happen through multiple different ways. I'll cover how MDMA functions in the brain and how it can lead to serotonin syndrome, as well as just using antidepressants generally, and genetic predispositions and environmental conditions which could lead to serotonin syndrome. So, just hopping into it, why am I making this video now? So, just to be straightforward, I guess I've been wanting to make this video for a while, and I've just been pretty lazy, didn't feel super motivated, and then something happened recently which kind of lit a fire under my butt to make this video, just because I think this topic will become more mainstream in the future. What do I mean by this? There was recently a press conference at the White House on April 18th where, of all people, Joe Rogan met with the president to sign an executive order to basically, and I'm just paraphrasing here, tell the FDA and DEA to calm down and let people research ibogaine. Ibogaine is a psychoactive compound which is found in the iboga plant, which is native to parts of Africa. And similar to other serotonergic agents like psilocybin or LSD or MDMA, also known as ecstasy, ibogaine is also psychedelic in the way that it alters your perception of reality. And the reason why Rogan was really excited about telling the FDA and DEA to loosen up and allow for research on this compound is there's already a decent amount of research showing that there's promise for ibogaine to be used medically. So there, I guess the one main study that was cited in the passing of this executive order was a study done by Stanford in the year 2024, where they took 30 veterans, and these veterans had TBIs or traumatic brain injuries, as well as post-traumatic stress disorder, and they were basically interested in seeing if they were given ibogaine, would their symptoms such as depression, would that go away just through one treatment with ibogaine? And what they saw were pretty crazy results of, I think it was anywhere between like 80 to 90% reduction of depressive behavior. Not like a total no more depression, but they saw some pretty exciting results in that these veterans who, went through some traumatic events were now able to not be so harsh on themselves and have to relive these moments over and over just through one session with ibogaine. And something else that ibogaine is known for is that people will fly out of the country to go to ibogaine retreats, where they will pay thousands, if not tens of thousands of dollars to consume ibogaine, go through this frenzy of a psychedelic experience to try and treat their substance use disorder. And commonly, a lot of people who go to these types of retreats are struggling with opiate addiction. And there's a lot of anecdotal evidence saying that ibogaine could be potentially helpful in treating those conditions, which I don't want to be the guy that is hating on this great thing, but ibogaine is still very far from becoming accepted medically as something that can be used to treat PTSD or substance use disorder, no matter how great um, of a story you've heard from somebody online. For example, I know Joe Rogan talks about it a lot. I don't care how many people he's met who have done ibogaine. We need to do clinical studies or clinical trials to actually see the utility of ibogaine to treat this type of stuff. But the one good thing that did come out of this executive order, which, may I add, there's not a lot of good that's come out of this administration so far, but I think this is maybe one of the things where I think that I could say that I am on the same page as the administration by basically telling the FDA and DEA to loosen up on research for psychedelic compounds. We could be very closely coming to a new age where psychedelic compounds are being used to treat certain mental health conditions. What do I mean by that? There is a company called Compass Pathways, which is very soon coming to the end of third stage clinical trials, which, if you don't know, after the third stage clinical trial, the company is basically good to start selling their product, and the FDA will just monitor them to make sure that there aren't any long term effects of that newly released drug, which they missed in the third stage clinical trial. So, Compass Pathways has they've produced psilocybin in a proprietary way, and are using it to help people with treatment resistant depression. And as I mentioned, they're close to the end of third stage clinical trials, meaning that it could be that by the end of 2026 or the start of 2027, we could see psychedelics starting to be used to treat mental health conditions, which is very exciting. There's another reason that I was excited to make this video, that being there's a new documentary released produced by Sam Harris, sorry, called Unraveling the Dream. And basically what happens in this video is that they talk about new theories about how the brain works, what the ego is, as well as talking about the history of counterculture movement in the US, how Nixon messed up everything and basically said LSD will scramble your chromosomes and you're a terrible person if you take it. And essentially, the the point of the video is trying to say that we might be coming up into this new revolution of, in the 70s we were pretty sure that psychedelics could have some great power medically, and that was crushed by Nixon, and we might just now be coming into this new era where we're able to research psychedelic compounds again. And there's many professionals in that video, for example faculty like Robin Carhart Harris, professor at the University of California, San Francisco, great institution, who has done a lot of research in this area and maybe more eloquently than me can explain to you why it's a great idea to use psychedelic compounds to treat mental health diseases and how it all relates back to the default mode network, a network in your brain, which I'll mention later on in this video. All that to say, though, is that before either of these things happened, I just didn't really seem. I don't know. I was just kind of lazy. I didn't want to make this video, but now that it seems like we're in this new age where potentially psychedelics could be used to treat mental health diseases, I think it's important to give the public a grasp as to how the serotonergic system works and have the same awe that somebody who is a neuroscientist can feel about how these compounds actually change your mind. So now let's transition into what the serotonergic system actually is. As I mentioned, serotonin is just one of many different types of neurotransmitter. Here I'm showing an image of just different types of small molecule neurotransmitter. And as you can see there, it shows that serotonin helps regulate your mood, your sleep cycle, as well as hunger. Many different types of neurotransmitters. Serotonin is just one of them. But looking at where it's produced in the brain, there's actually just like a small amount of neurons in the brain that are able to produce serotonin, all of them being near the brainstem, kind of in the less evolved portion of the brain. If you're thinking about what has evolved more recently, think about the prefrontal cortex up near the front of your brain. But this is kind of an older area of the brain. And I think the most interesting part of the serotonergic system is that of those red clumps that you see there, uh, those red clumps, those are the only neurons in your brain which are capable of synthesizing serotonin, which can then be used to communicate with other neurons. So these red blobs are where the serotonin is produced, and the red arrows are, in theory, where the serotonin could go to influence other neurons. And the craziest part about this is that there's roughly only 250,000 to 500,000 serotonergic neurons in your brain. And it might not sound like a crazy number, but when you think about the total amount of neurons in your brain, which reaches somewhere near the 100 billion neuron range, you see that serotonergic neurons really are in the minority and are almost insignificant in the total count of neurons in your brain. And yet that is the system that we choose to target our therapies towards to treat mental health conditions. And when you take substances like psilocybin, they produce such a profound change in the way that you view reality, even though the serotonergic system is such a negligible amount of your brain. And that is something that truly stuns me and hopefully impresses you at least a little bit as well. And just comparing this with maybe another neurotransmitter, for example, GABA, which is the resident inhibitory neurotransmitter in the brain. If you were just looking at an image of distribution of different types of GABA in the brain, you can see it's pretty readily distributed across the brain when you compare it to serotonin receptors. All that just to say, there really isn't a lot of serotonin, and somehow it plays a massive role in the way that your mood is formed, as well as the way you think about yourself. But that's all something we're going to cover later. Now we're going to transition into how is it that compounds like psilocybin modulate your brain. Psilocybin famously interacts with the serotonergic system. So this compound somehow interacts with a negligible amount of neurons in your brain and produces this crazy experience, which we all know. But what is actually happening in the brain when psilocybin is consumed? We're going to have to go and look at the MRI data, which is in this paper by Dr. Joshua Siegel and many, many others, um, where we're looking at this how psilocybin desynchronizes the human brain. So basically, what happened in this study is that Dr. Siegel and all of the others in this study, what they did is they took participants and they did fMRIs of these people brains. So what an fMRI does is it's looking at the oxygenation of the blood in your brain. And just to remind you, I think everybody knows the mitochondria is the powerhouse of the cell. But what people don't remember, and what I'll remind you of now, is that once the electrons are done going through the electron transport chain, they need to be collected by oxygen so you just don't have free electrons roaming around the mitochondria. Meaning, essentially, if you have more oxygen in a specific region, you could safely assume that that region has more activity going on. That part needs more energy. It needs to produce more ATP, meaning it produces more lone pair electrons, which need oxygen so that the electrons can get onto the oxygen and then the whole cycle can be complete and the body's made energy and all that good stuff. Basically, when you have a brain region with more oxygen, it basically tells you that brain region is more active. And if there's less oxygen, that brain region is less active. And you can use this to see if neurons are more or less active, as well as seeing who they're talking to, which neurons are actually using their energy to communicate with. And so, what they are using these MRIs for is to look at the brain before, during, and then after these people had this experience with psilocybin. So during, there actually were participants who agreed to take psilocybin, and then while they're peaking, they get into an MRI machine, which, thank you for your service because that seems absolutely terrifying. But let's just go into the data of what they found here. So, looking at just this first figure, we're going to be looking at a change in functional connectivity, or FC change. And this is essentially a measure of how quote unquote promiscuous a neuron is being. Or, in other words, if there is low FC change, that means that that neuron is not doing anything out of the ordinary. It's talking to the same 4 or 5 people that it normally does. Whereas a high FC change means that this neuron is now talking to other neurons, which normally it doesn't send any signals to, basically showing that this brain is kind of in this new novel, quote unquote, desynchronized state where neurons are able to talk to other regions of the brain where they normally don't have a chance to communicate with. And what we're seeing in this figure, a low FC change would be like a black color, and the very high FC change would be a deep red. You can see the prefrontal cortex lighting up very deep red. Prefrontal cortex is associated with um, like high level planning, critical thinking, emotional regulation, stuff like that. And you can see that the prefrontal cortex is lighting up deep red, meaning that all of those neurons, which normally go into regulating your emotions and planning about the future, are now talking to neurons which they normally don't talk to anymore. There's also many other brain regions which saw a high change in functional connectivity. But one of the most important things in the study that they noted is the change in connectivity of the default mode network, or DMN. And this is basically a network of regions in the brain. And I'm showing this picture here, just showing that there's some parts of the prefrontal cortex, as well as some of the basal ganglia, as well as cerebellum, which are all involved in creating your image of yourself in your head. So, whenever you're thinking of, for example, you have an internal monologue going on, you're talking to yourself, or you're thinking about the past or the future, or you're just not you're not having any sensory information coming in. this is when your brain is humming along in its default mode network, and those neurons are kind of going in the background when there's nothing else to do. Once you've collected a lot of sensory information throughout the day, let's say you go on a bike ride, you fall and you hit your knee, and then you go to a new food spot and you find this new burger that you really like. One At the end of the day, you're going to bed and you're thinking about what happened that day. You're like, oh wow, like my knee really does hurt, but that sandwich was really great. But also, I think I need to set aside some time because I have some work that I got to do and something tomorrow. All of that internal thought process is your default mode network humming along. And if you still aren't really sure what I'm talking about when I say the default mode network, is kind of the opposite of meditation, in the sense that when you're meditating, at least when I try to meditate, the only thing I try to do is just close my eyes, deep breaths, and not to think of anything. So when you're deep in meditation, it has been shown that the default mode network of experienced meditators goes quiet. Whereas, for example, with me, it is screaming very loud and it makes me not able to sit still for 5 minutes because I'm constantly talking to myself. But interlude aside, that is the default mode network. And what they saw in these MRI images of people when they were taking psilocybin is that regions associated with the default mode network had a change in functional connectivity, basically meaning that the default mode network, which is like all reliable when nothing else is going on, that's humming along. Now, if you're taking psilocybin, the default mode network is no longer acting in its normal way, in the way in which you think about yourself and generate your own reality of how you perceive the world, and it's working in a different way because psilocybin is in the brain. Specifically looking at these two brain regions, or three, you have the hippocampus and the basal ganglia, you have the thalamus and cerebellum. I won't talk about what each of these do and how they contribute to the default mode network. But all you need to know is that a bunch of different regions involved in the default mode network had a different functional connectivity when people took psilocybin, which is very interesting and kind of makes sense when you consider the types of stuff that people tell you about those um, trip experiences. And that once you take enough of a psychedelic, you start losing your sense of self. You have ego death, basically, where you aren't able to think about yourself anymore, and your default mode network kind of dissolves from its natural ordered state into a desynchronized state, if you will. So, yeah, just looking at, I guess now we're going to just wrap up this paper, but as I mentioned, the default mode network, this is a pathway in your brain which had the highest change in functional connectivity of a variety of other networks in your brain, which work to generate your sense of what is going on around you. That was the one that was altered the most when patients took psilocybin and when they observed the changes in connectivity of their brain using the MRIs. Before we move on, just I guess a couple of more tidbits to look at, and just to, I guess, conceptualize where all of this fits in with current knowledge. So, just looking at, I guess, the control conditions, because I just showed you the change in functional connectivity when people took psilocybin. Now, looking at, these are the scans that they took before people took psilocybin and went into the MRI. And what you can see is that there is a change in connectivity day to day, basically meaning that if a person came in one day and then another day, both times sober before they took psilocybin, their brain looked um, relatively similar in the sense of which neurons were talking to which neurons. So there was a pretty drastic difference in the connectivity of the brain sober and then while taking psilocybin. Something interesting that they noted as well is that this effect of functional connectivity change seemed to persist for 3 weeks in these regions of the brain heavily associated with the default mode network. So, as I mentioned before, the hippocampus, we're looking at this first acute change, which is basically when they took the psilocybin originally, what type of functional connectivity change did they have, and then looking at 3 weeks post, seeing if there is still that change in functional connectivity after for 3 weeks not taking any psilocybin. And as you can see, this is something else that I forgot to mention, but there's only 7 people in this study. So similar to the Ibogaine study by Stanford that I mentioned earlier. I mean, the Stanford study had 30 people, which there's some statistical significance to be had here. But with 7 people, it is difficult to have that many MRI sessions with that many patients. But um, this does limit the scope of the results a little bit, just in terms of there isn't as much to go off of, and this is certainly a good jumping off point. But I think it is decently conclusive enough to reason that under the influence of psilocybin, at least, there's profound changes to the way that the brain talks to itself. And we can see that here with these results showing that 3 weeks post this experience, the default mode network is still acting in a different way. And if you watch that video, Unraveling Your Dreams by Sam Harris, default mode network, uh, it is kind of in a state of, it is one of the more higher order things in your brain, in that it doesn't want to change from equilibrium. So even potentially small changes in the default mode network could lead somebody to having a radically altered view of themselves, and could lead to them getting out of these and could lead them to view themselves as a different person as a function of the default mode network changing its signaling in slight way, in which that patients may no longer have treatment resistant depression, for example, even after only one dose of using psilocybin, which is something that has been seen by compass pathways. Now that we've already talked about, I guess, how psilocybin can profoundly change the serotonergic system, let's talk about some of the hazards of targeting the serotonergic system. That being, since it is such a small system in the brain, there's roughly only 500,000 neurons at most who produce serotonin. Even small perturbations in the amount of serotonin in your brain can lead to serotonin syndrome, which, as I mentioned, leads to autonomic dysfunction such as dilated pupils, diaphoresis or sweating, as well as trouble sleeping, um, trouble controlling heart rate, as well as blood pressure. There's very many things that can go wrong with serotonin syndrome, and there's a scale of severity there, where you can have something where is not super severe case of serotonin syndrome, and there's a very severe case where you could end up having seizures, for example. But I won't really go into the clinical indications so much. But if you're interested more in the clinical stuff, I'd highly recommend checking out video on serotonin syndrome by Strong Medicine, who, if you're more in the clinical sense of mind, that was probably better for you. But now we're going to talk about what are some cases in which you could develop serotonin syndrome, and what is happening at the molecular level allowing that to happen. So we're going to go back to our serotonergic neurons, which are in the red clumps there. And these are the only neurons in your brain which will produce serotonin. So, looking at the serotonergic neuron here, we're looking at the presynaptic neuron here in yellow. What it does is that it produces serotonin from tryptophan, which is an essential amino acid which we get from our diet. And I think classically people would know about tryptophan because it is in high abundance in turkeys, and also I think it leads to being tired So I think that is maybe the one way in which people have heard of tryptophan. But anyway, it's one of 20 amino acids which is used to synthesize serotonin through the use of two enzymes here shown as TPH and DDC. So once you have the 5-HT or 5-hydroxytryptamine, then that serotonin gets packaged into a vesicle via VMAT, and then once you have a bunch of serotonin packed into this vesicle, you can eventually get to the point where this vesicle binds and merges with the presynaptic membrane, and all of the serotonin that was inside the vesicle now becomes part of the outside of the cell and then can flow and bind to the postsynaptic neurons, wherever those may be, whether it be the prefrontal cortex, the hippocampus, the thalamus, or the cerebellum. And so once serotonin is in the synaptic cleft, it can then bind to all those neurons postsynaptically. And then what happens is after the serotonin signaling has finished, then the serotonin reuptake transporter or SERT can take serotonin and bring it back into the neuron. Once serotonin is back into the presynaptic neuron or the neuron that originally had synthesized it, one of two things can happen. It can either go back into a vesicle via VMAT so that it can go through another firing process again, or it can be degraded by monoamine oxidase. And here is where I'm going to introduce two types of antidepressants. One of them is SSRIs or selective serotonin reuptake inhibitors. So, SSRIs act on serotonin reuptake transporters or SERT. So, an SSRI would bind that green channel there and prevent serotonin from getting back into the presynaptic neuron. What this does is that now when a neuron fires naturally, then the serotonin will go onto the postsynaptic neuron. That's basically the same, but once the serotonin has bound and it's ready to go back into the neuron, less of it will be able to do so because the SSRI is blocking SERT from taking the serotonin back into the neuron, making the serotonin free to keep binding with serotonin receptors and increasing serotonin function. Another way in which increased serotonergic activity can be caused is through the use of monoamine oxidase inhibitors, or MAO inhibitors for short. And MAO inhibitors are inhibiting the enzyme monoamine oxidase, which breaks down serotonin normally, leading to higher amounts of serotonin within the neuron, meaning more can be packaged into vesicles and more serotonin will be put onto those downstream neurons because it can't be degraded as readily. Now, let's see how MDMA works. MDMA is thought to be kind of psychedelic adjacent, but it is also a serotonergic agent similar to psilocybin and LSD in the sense that it enhances the activity of the serotonergic system. And let me just say, MDMA is truly kind of an evil molecule in the sense that if you're taking an SSRI or a MAO inhibitor in conjunction with MDMA, also known as ecstasy, I really don't envy you because that is a really bad place to be in. Even MDMA on its own is so potent in the way that it acts. I mean, obviously, it's dependent on dose, but still, just the way that it acts in the brain is so nefarious that it's kind of truly beautiful in a kind of crooked way. So just looking at the pathology of what happens with MDMA when it gets ingested. First off, what happens? MDMA hijacks SERT or the serotonin reuptake transporter, if we're looking at number one on the diagram here, and it goes into the presynaptic neuron. Once in there, what MDMA does is that it blocks VMAT. So all of the serotonin that is made by the presynaptic neuron is unable to make it into vesicles so that those vesicles can merge with the presynaptic membrane and cause I guess the transfer of information from the presynaptic neuron to the postsynaptic neuron. And then what MDMA also does is not only does it prevent serotonin from going into the vesicles, if we're looking at number three, it also blocks monoamine oxidase, meaning that not only will serotonin not go into vesicles, it also won't be degraded once it gets back into the post presynaptic neuron. Sorry. This means that there's a lot of serotonin just freely floating in the axon terminal of the presynaptic neuron. Lastly, and probably the craziest part of MDMA, is that it also will bind to the serotonin reuptake transporter and reverse flow of serotonin. So instead of taking serotonin from the outside and taking it back in, it's going to push all of the serotonin that's being made by the neuron out onto that descending neuron below it. Um, and as you can see, if we look back in this image here, if we also prevent the serotonin reuptake transporter using an SSRI, there's even more ways in which the serotonin is not able to make it back into the neuron and keeps hanging out in the synaptic cleft. And same thing with MAO inhibitors. It prevents the breakdown of serotonin so that there's just so much of it where your brain kind of starts freaking out that there's so much serotonin in your body, where you start having autonomic processes, as I mentioned earlier, not going the way they're supposed to anymore just because there's such an overload of serotonin there. So, if you are taking antidepressants, specifically MAO inhibitors or SSRIs, I'd highly recommend not taking MDMA at the same time. I'm not a doctor. I don't even have a doctorate. I'm just an undergrad guy, but I that is my advice. Don't take medical advice from me, but I would say if that is something you're willing to do, you could potentially meet fatal consequences because your body isn't meant to be able to handle that much serotonin. Which, by the way, another thing about serotonin syndrome that's crazy to me is that um, it's kind of unthinkable that serotonin syndrome would happen to anybody, um, even like 200 years ago, just through the fact that there's so few known of natural ways to influence serotonergic activity in the brain, where you kind of need Western medicine to influence the serotonergic system to be able to create serotonin syndrome. So this is a very recent, I guess, condition that sprouted up. But um the interesting thing about serotonin syndrome and how it ties back to compass pathways is you would think that when you have psilocin and LSD, which also mimic serotonin in the brain, um, they act differently than MDMA, which MDMA indirectly increases serotonin levels in the brain, whereas LSD and psilocybin basically just mimic it. And looking at this image here, you can see that their structures are very similar. So you can see why, I guess, the serotonin receptor would think that serotonin is bound when you have LSD or psilocybin floating around. But the interesting thing is that compass pathways have shown that of the people who were in their trials, a large majority of them were taking SSRIs or other antidepressants at the same time, which begs the question, why is it that you don't get serotonin syndrome with psilocybin, but you do with LSD? And you do with MDMA. Sorry, oh, this is getting to be a long video, and I'm losing my mind right now. But in essence, the reason for that is that um, LSD and psilocybin, uh, they are both only partial agonists at the serotonin receptor, meaning that when serotonin binds, there's some sort of effect that happens in that postsynaptic neuron. And when psilocybin or LSD binds, that same effect happens, but to a lesser intensity, meaning that potentially because of that, there isn't enough shift in serotonergic activity where you could get serotonin syndrome by mixing um, psilocybin or LSD with SSRIs or MAO inhibitors. Although I will say this isn't medical advice. I wouldn't say that you're good to just do that stuff just because current clinical studies, it seems that it's okay. I would consult a doctor because, yet again, I am not a medical doctor or even have a doctorate, so I would not know. But lastly, just to round off this video, I talked about how serotonin syndrome could be had by mixing antidepressants like SSRIs or MAO inhibitors with MDMA, which obviously is an illicit substance, which, not everybody is going to want to try an illicit substance. So is serotonin syndrome only going to happen if you mix drugs like that? There is actually a possibility in which you are a goody two shoes and don't want to get into that. You don't want to get into illicit substances and still get to experience, not get to in like a good way. Like, I hope nobody has to experience serotonin syndrome in their life, but there is a way in which you're able to get serotonin syndrome not through the combination of antidepressants and illicit drugs, that being potentially through drug interactions or even potentially just through changes or differences between individual people's drug metabolism. So we're just going to talk about drug metabolism for just a little bit here because I think it's interesting. So the main way the drugs are metabolized in your body is something called first pass metabolism, where basically if you take a drug orally, it is subject to all of the enzymes in your body as well as many other things which try and break it down so that it is no longer usable by the time that it gets to the blood. One of those such enzymes is called cytochrome P450, which hangs out in the liver, and you can see the many different types of cytochrome P450 here and all of the different types of drugs which these types of enzymes, um, they don't degrade them but they modify them in a way such that they are no longer useful. Looking at the active site of one of these cytochrome P450 enzymes, you can see there's iron core in the center there, and you have this, um, I guess, what do we call the prosthetic group? I don't know, but it's not part of the protein, but there's this heme group. I think it might be a heme group that is called. But you have this group that is holding this iron atom, which catalyzes a reaction, which I guess happens to all drugs that pass through the liver, in that you add specific elements onto these compounds in your blood, hoping that it makes it such that these compounds don't interact with other parts of your body. So, looking at the way in which cytochrome P450 works, um, what you see here, starting out, you have this R group connected to a hydrogen. So, let's, I guess, in terms of trying to make this more concrete for people, because I know some may not be very excited to take or have never taken OChem. Let's just say that we're thinking about sertraline, brand name Zoloft. You take some Zoloft and you swallow it, and then once it gets to your liver, what is happening? Well, a cytochrome P450 enzyme, its whole goal is to make it such that compounds that are in the blood stay in the blood. And how do you do that? You add a bunch of electronegative stuff onto whatever it is that you found in the blood. And the reason that you're doing this is you don't want to find something in the blood and let it just hang out and potentially be able to get inside one of your cells. You want it to stay in the blood and eventually get excreted out through your urine. And the best way to do that is to add electronegative elements such as oxygen, for example, which can make hydrogen bonds with water and can stabilize a compound such that it doesn't really want to go inside of a cell. It wants to stay in the bloodstream and doesn't actually have a chance to exert an effect. So, looking at sertraline, it has hydrogen, which if the sertraline got in conjunction with this cytochrome P450 enzyme, what would happen is that this enzyme will attach an oxygen and put it in between the carbon and hydrogen on the citrulline, altering the structure of citrulline, not only making it so that it has a different activity, potentially different function in your body, it also makes it such that it's harder for it now to pass through um, lipid membrane bilayers since it is more polar. And just looking at this last image here, I guess, of citrulline, what I was mentioning is this citrulline, there's many different types of cytochrome P450 enzymes, as well as monoamine oxidase, will also degrade citrulline into many different byproducts. And essentially, when a physician is prescribing you Zoloft, what they have to take into account is hopefully that you have a normal metabolism, and they're banking on the fact that the drug will be degraded at a specific rate such that you have some specific active amount in your body. For example, I don't know the average dose of Zoloft, but let's just say it's 50 milligrams. If you take 50 milligrams of Zoloft, it's pretty safe to say that most of it is lost as byproduct, and maybe only 2 milligrams of that is actually active and able to do work as an SSRI in your brain. However, one way in which you can get serotonin syndrome, in which you don't take any illicit substances, is that cytochrome P450. There's many different types of it, as I mentioned earlier, and this is controlled by genetics, and many different people have different varying amounts of this enzyme in different quantities, meaning that there are certain people who for certain medications will degrade it at a faster or slower speed, meaning that, for example, let's say I got a Zoloft medication and the standard um dose is 50 milligrams, and you'd expect 2 milligrams of that to be active once you go through first pass metabolism. If I have a deficiency in the cytochrome P450 enzyme, which is responsible for degrading Zoloft, there's a chance that once I take the 50 milligrams, I might end up with 4 milligrams of Zoloft in my bloodstream, meaning that I have an increased amount of drug action in my body, not because I've done anything wrong, just because my genetics make it such that I'm worse at degrading Zoloft than other people. Another interesting, and I think probably my, I wouldn't say it's my favorite fact about serotonin syndrome, but it is that if you are taking some SSRIs, there is a chance that you will not be able to drink grapefruit or eat grapefruit anymore, just because there is a naturally occurring compound in grapefruit which blocks the degradation of those SSRIs. Um, yeah, that is all, I guess, for serotonin syndrome. I hope that it's been informative and hopefully changes your mind if you are on antidepressants and want to try ecstasy. I'd highly not recommend that. For that matter, I don't recommend doing any illicit substances. I think that would not be very smart for you to do. But I hope that understanding how they function gives you a greater appreciation for how they work and potentially give you some more background, given that they might become a more common use in medicine in the coming years, given this, I guess, recent executive order. But we'll all see how it pans out. I'm not really a betting man, but if I was, I would definitely say I would see psychedelics used in medicine in the next 5 to 10 years. I hope this video gave you some insight and maybe an appreciation, or give you maybe the same awe that I have for the serotonergic system in the human brain. Um, but yeah, as always, hope you've enjoyed, and I will see you in the next one.